Hello and welcome to this video on the role protein plays in brewing. For the most part, this video will focus on beer, but there is relevance to wine and other fermentations. Proteins are present in just about all living things. This includes the grains, fruit and other material used in fermentations. Even the yeast used to create fermentations has to have protein present. That means protein will play a role in whatever you try to make. Generally, the role proteins play is either an outright flavour, or to be broken down into smaller constituent parts called amino acids. These contribute to flavour and other parts. Without protein, some of the complexities of flavour are impossible. At the same time, protein can also be a curse when off flavours are made. Protein, if it is intact and as a consequence of fermentation enough is present, results in things like opacity, viscousness, and the stability of the head in your beer. The length of the different proteins in beer can have big effects on this. Some of these are benefits, others are detriments. Shorter proteins will find their way into the wort. This is because they come through the different processes that occur relatively intact. In doing so, they contribute to the body and aid in the bubbliness of the head in beer. What you want to avoid are long chains of protein. These can result in what we would consider haze or a chill haze in the beer. There are two primary proteins. Both of these are albumins associated with the beer formation. Protein Z and lipid transfer protein. These are needed to get good foam and head in your beer. Without these, you're also going to have difficulties with getting the body or the density and mouthfeel of your beer right. It's somewhat fortunate, if not likely a result of the development of beer, that during the beer brewing process, proteins are broken down from those long chains into short monomeric proteins. In the end, about 70% of what you find in the mash after it has been put through the lautering process are going to be small monomeric proteins. The rest are large. The percentage of large polymeric proteins, these are the very long chains, in your mash, when it is malted, goes from 27% in raw barley to 20%. This is the problem area. You need to get that number as low as possible before doing anything else. Even if you were going to rest your mashing process, this isn't going to change that number hugely. The difference is that lautering will ensure that any proteins that are left in the grain are only those that can't be extracted, and largely those that can't be extracted are large proteins that are adverse to the beer flavour, taste and mouthfeel. It's noteworthy that you do need protein for some beer in particular. There's notable amounts of proline or hydroxyproline. These can then have an effect on the colour. This is because, to a certain extent, proteins affect the absorbance, and this is going to affect the change of colour from alpha amino acids. Alpha amino acids being one of the two kinds, alpha and beta. For the most part, by ensuring that you get primarily proline and hydroxyproline, you get a change in colour, generally being a reduction in colour. Looking at this another way, you can consider that most of your protein in beer will come from barley that has been malted. This is because barley makes up the vast majority of grains used in beer. Contrasting this with grapes, for instance, grape protein content has been known about for 2 centuries. We know that they do contain a number of different kinds of proteins, but they're not especially high, especially by comparison to other fruits. This is useful in some ways, notably in that you primarily get sugars, some flavour compounds, and a little bit of protein, but you don't need to worry about it anywhere near as much. The same proteins found in grapes tend to also be very soluble and very short. This means you get very simple, very low weight, proteins that tend to not have negative effects. This is further assisted by the fact that about half of these proteins can easily be bound to phenolics. Phenolics are one of the more active chemicals within red wines particularly that give them their sometimes bitter flavour and also their colour. In white wine, it's the cause of haze, or at least one of the more common ones. Because of the difference in the kinds of phenolics found and the quantities of protein, you'll find that red wines don't demonstrate the same degree of protein precipitation and consequential haze as red wines do. And this is one of those considerations. Where it's so very important in beer, in wine, especially white wine, it can have an adverse effect. This doesn't of course mean that protein isn't a concern at all. Different wines will lead to different kinds of opacity or turbidity, when unfortunately they're exposed to differing temperatures. Generally speaking, the hotter the temperature in which the wine is fermented, the more likely you are to observe turbidity. This is something similar to what would occur in a fluid culture when you get something growing in it. In this case, it's not due to a pathogen or other infection, but rather due to the fact that the protein causes various connections to other molecules, and these begin to coagulate. Or at least, that would be the general thought at first. However, given the same proteins in different wines, it would appear that proteins themselves are not necessarily the big factor to play. Rather, non-protein factors come into play with protein, and they simply act as the intermediary. This means that under different circumstances with exactly the same protein profile, in different wines you may see drastically different degrees of development of haze and or turbidity. This process is generated largely when the grapes are processed to create grape juice. The juice and the crushing of the grapes puts some protein in, but since you're not using the skin anywhere near as much, you get much less of the protein, which makes up a large portion of the outer part of the grape and the seeds within it. All of this is filtered out. Compare that to the mashing process for malted barley. The entire barley grain is processed and you get a great deal of the outer part of the grain plus the inside, and both have differing degrees of what we consider useful and detrimental protein in them. Grapes just happen to have far less of that area used and far less extracted. The process of turning grape juice into wine also involves fewer steps and less extraction of other material. The heating, mashing, and other steps involved in making beer will extract some of these, but thankfully the steps used in beer tend to minimize the amount of unwanted protein that is present. Thank you for watching this video. If you found it interesting, consider liking, sharing, and subscribing. Please post any comments, questions, or suggestions that you have below.